So we have package demo, which will be presented by Marcel Ramos. He is a core Bioconductor member. So Marcel, please go ahead.
Thank you, Mikhail. So thanks to everyone for coming. I prepared just a small set of slides to orient everyone on how the workshop will go about. It's a package demo, but I want it to be more interactive. So if you'd like to access these slides, which have some relevant links, or you can just follow along, this is the URL for these slides. The package down website that has been rendered, you can see it here, waldronlab.io, multi-assay workshop. And to launch the workshop instance, if you can, it's workshop.bioconductor.org. It should be working now, hopefully. We had some issue at Vince's demo, so hopefully ours will go a little bit more smoothly. So it's a little tricky because when you click the launch button, you have to wait and see that there's a link that shows up, so it's not very prominent, and we're going to work on that. But just look out for the click here to open link at the page. And once you're inside the instance, you can do help package equals multi-assay workshop, and that will pull up the documentation within the RStudio instance. So for those of you that don't know me, I'm Marcel Ramos. I've been working with Bioconductor for about 7 to 8 years now. I mainly develop software for integrating multi-omics data, but I also work on Biopsy Manager and other software that needs help in Bioconductor. So I guess I'm the utility baseball player in Bioconductor. So so to get started, I started my own instance here so that you can't copy my URL, or you can, but then you'll just be on your computer. But what I've done is just spin up the Docker image. You don't have to do that. You should be using the Galaxy workshop here, and you'll be able to see our package demo link on the left, which has package demo multi-assay experiment right there. If you can see should be able to see my cursor. Okay. So we'll while our instance is populating and coming up, I will go over just some of the basics of what I want to present here. What I'll do here is maybe go over the packages that we are using for the workshop. And by a show of hands, how many people are familiar with multi-assay experiment already? Okay, that's good. There's a couple. And so has anyone heard of curatedTCGAdata? Okay, so we have new customers. Okay. So multi-assay experiment is a infrastructure package in Bioconductor that takes all of these different multi-omics data representations, like mutation data, copy number data, methylation data, and puts it all in one container so that it's easier to coordinate and subset your data. In terms of methods, we don't have, multi-assay experiment mainly focuses on the data representation more so than the methods. I let others work on the methods, and they can certainly extend multi-assay experiment to provide PCA methods for between multi-omics data types. So multi-assay experiment is that container infrastructure package. curatedTCGAdata is our it comes out of the pipeline that we've been working on, which takes some of the legacy TCGA data. I say legacy now because it's been quite a few years since they've updated the broad fire hose data pipeline, and that's the origin of the data in curatedTCGAdata. For the newer harmonized data, if you're looking for the newer harmonized data, I would recommend the Genomic Data Commons package. So the curatedTCGAdata makes it really easy to pull in data from the TCGA project and represent it in a multi-assay experiment class structure. And more recently, I worked on this Terra TCGAdata. So if you've used the Terra platform, this would allow you to take some of the instances, or I think they're called workspaces on Terra, and use the data from those workspaces to import as multi-assay experiments from the Terra platform. So I think, so this package works more on the data that comes with the workspaces. There are two different types of workspaces on Terra that work with TCGAdata. This package works on the data that is shipped with the workspace. It doesn't really work with the data model. But if you need more details, just ask me after the workshop. And then I've also worked on the cBioPortal data package, and this package allows you to import cBioPortal data. So there are about 306 published in cBioPortal, and what this package does is accesses the API on through the website, pulls down the data, and provides a multi-assay experiment for you. And there are two main ways of using the package, but I won't go into the details just yet. And TCGAutils is a helper package that allows you to work with the data that you get from curatedTCGAdata to sort of only select tumor samples, for example, or change some of the features or transform the features in the multi-assay experiment that you get from curatedTCGAdata. So it has a number of utility functions that you might find useful. It's a little bit scattered in terms of the organization because it's a utility package, so it has varying functionality in the package. And then more recently, we've worked on the SingleCellMultiModal package along with collaborators in the University of Padova, Dario Vigeli, and Kelly Axenrode. The SingleCellMultiModal provides single cell data from different studies as an attempt to obtain benchmarking with these relatively new technologies. And then this TENxIO package is one of my more recent efforts that attempt to import TENx files as Bioconductor data classes. So I structured it in a way that it takes a file and gives it a class, and then provides an import method so that you can represent the data as a Bioconductor class. So, so it should make it easy for you to import TENx data files quite quickly. And I think we are working on some import methods for the Visium format. So that is currently in the works. And so with that, I will start to describe our main data representation, which is multi-assay experiment. And there are three main components in the class, and maybe I should make this bigger. Or maybe, so on the left, bottom left, you'll see that there's the experiments components of multi-assay experiment. So this is a separate class to handle all of the different experiments that you can put into a multi-assay experiment. And it's pretty flexible. It's a sort of list-like structure that allows you to just put together all of these different assays that may have varying dimensions. So you may have a varying number of features and a variable number of samples in the experiment. So, and the way you would pull out the experiment list class representation is with this it's called an accessor function. So experiments and the multi-assay experiment. So you would do that, and that would give you the experiment list class. On the top right, we have the call data. So if you're familiar with SummarizedExperiment, this is the same concept. You have the phenotypic information of the patients in the data, and every row in this data set is a particular patient. So these green lines that you see here are can represent the trace of one patient. So that patient could have two samples in one of the assays. It may have missing samples in another assay, and so on. So you can see that they can have a variable number of samples and even replicate samples in assays in the multi-assay experiment. And then on the bottom right, we don't really interact too much with the sample map, but this is just to show you that everything is sort of tracked in a multi-assay experiment with this sample map representation. And it's essentially a graph representation of all the data within the multi-assay experiment. So there are three main columns: the assay name, so all of these assays should have a name, the patient ID or barcode, or the observation, which is usually like a sample ID. So those are the three columns in that sample map. And I think I went over some of these, but so I'm going to skip over the points here, but feel free to go through those and take a look. So maybe I should show you the schematic of how these things can come together in our little ecosystem of packages that work with TCGAdata. So we had the TCGA main data set. It went through the GDAC Firehose pipeline, and we mainly obtain our data from this package called RTCGA Toolbox. And then it went through the pipeline, so we did some pre-processing steps and some curation. One of those curation projects that Levi Waldron had in the lab was to add subtype information to not every, but a handful of cancer types in TCGA. So that was added in this pipeline, and then it gets modeled into a multi-assay experiment, and eventually you get these three packages. And then we have another package called the RaggedExperiment package, which we published not too long ago in May. And this package allows you to represent copy number data and mutation data in a way that multi-assay experiment can understand. And the main representations that multi-assay experiment understands are tabular format, so they're rectangular shaped data sets like matrices, etc. So, and I would consider SummarizedExperiment rectangular, right? Because you have your expression matrix. So the operations that you would do on a matrix apply to a summarized experiment. So you're familiar with SummarizedExperiment? Who knows, or who is familiar with SummarizedExperiment? Okay, yeah. So So let's go back here. I don't know if our online audience can see my extra window, but hopefully they can. So summarized experiment is sort of what we modeled multi-assay experiment from. And you have your expression matrices, which can have a third dimension. So you could have more than one matrix. You have the features on the left-hand side. So these are gene IDs, and the samples, the call data correspond to the patient. So similar concept. And we also have,
If people have trouble hearing you, can you speak up a little bit?
Yeah.
Not here, but through your microphone.
Yeah.
It's probably.
Okay, thanks. So there's also this metadata slot, which is a list type of class here. It's unstructured, so it's represented as something like that. So very similar to multi-assay experiment, and that's the basic structure of the summarized experiment. So just a little bit more about RaggedExperiment. We have we've actually updated this diagram in the paper, so I would point you to the paper. And let me, maybe I think I have it up here. Okay. So RaggedExperiment, in this example, we have a set of ranges that are, we have ranges at the top here. So this is represented by these whiskers. And every row in this sort of schematic here is a sample, right? So you can have a disparate number of measurements per sample, right? And what we want to do is to take this, what is usually modeled as a G-ranges list, which is a list version of a genomic ranges, which hopefully you're familiar with. We take this G-ranges list representation and sort of make it into a matrix-like format, depending on how you want to combine the ranges. So the easiest way to combine them is to just sort of stack them on the diagonal here. So it's a little bit sparse that way, and it might not be the way you want to combine these ranges. But you can also take, sort of look at the range information and see if any of those ranges match up, then you can sort of juxtapose them and save some space rather than having a sparse assay. So these gray boxes represent matrices, and that's what we're trying to do. We're taking the list representation and converting them to a matrix so that multi-assay can work with those. There's also the Q-reduce assay operation, which takes into account your region of interest and combines the measurements. And typically, these could be scores or anything in the metadata column. Usually, it's a numeric type of score. And you can combine these scores based on a region of interest. And then this joint assay allows you to sort of break all of these ranges up based on all of the other ranges in the original representation. So you have disjoint regions and measurements for each sample. So that's what RaggedExperiment does. It takes that list representation and converts it into a matrix so that you can use it in multi-assay experiment. All right, so maybe let's take a look and see if you've gotten, if you were able to get your workshop running. And if you don't have an active workshop, or if you're just uh, following along on the screen, then that's fine too. But hopefully your workshops are running now. Okay, I see a few nods, so that's good. Okay. We have a subclass of multi-assay experiment, which sort of cuts off the loose strings here. If you have, if you're only interested in a set of a set of patients that have measurements across all of the assays, you can convert into this matched assay experiment, and that will drop any patients that don't have an assay in all of the recorded assays. So it's a convenience subclass that makes like having just one set of patients with a measurement across all of the assays. So it makes it easy. So let's try the mini ACC demo. And if you weren't able to get this vignette up on RStudio, you can do, as I said earlier, help package equals multi-assay workshop. Right, if you do that, that would pull up this, and then you can go into package vignettes and other documentation. And what we're looking at is this one, the integrating multi-omics data with multi-assay experiment vignettes. Right. Okay, so I know there are a lot of classes to work here, but I think we'll just mainly focus on the multi-assay experiment. But the idea for multi-assay experiment is to sort of save you from thinking about all of the different classes in Bioconductor and allow you to coordinate all of your samples into one object. And say if you're only looking for a particular phenotype in your call data, you can subset your multi-assay experiment by that phenotype. So why don't we start with the mini ACC demo. So this is a small data set that comes shipped with the package to give you an idea of what the a small multi-assay experiment looks like. So you can do data mini ACC, and you can do package equals multi-assay experiment. So it does take a little bit because the multi-assay experiment hasn't been loaded, but should pop up in a second. Maybe I'll do mini ACC, just print it out. All right, so actually this happens when you don't load the package first. So I'll do library multi-assay experiment first, and then do data mini ACC. Yeah, so the time that we're waiting just means that I should update my computer because it has quite a few years behind it. So once you are able to load the mini ACC dataset, this is adenocortical carcinoma, and you can see that we have RNA-seq data as well as GISTIC data, protein array data, mutations, and microRNA. So you have these different data types in one container, and their representation is noted here in the show method. So you see we have a few summarized experiments and even a matrix. So it says click here, so let's see what happens. Okay, yeah, it wasn't going to work. So what you can do to open up the Shiny tutorial, there is a folder in your files, which is ints, tutorials, and we'll do multi-assay experiment, and we'll open up this exercise multi-assay R&D, and then we're going to click on run document. So this is just a tutorial to show you what the different components are and how you would build them separately and then put them together in the last step to generate a multi-assay experiment. And when you click run document, this runs this, it's a Shiny document here that runs the tutorial. So let's wait for that to load a bit. Okay, thanks. For the online audience, if you see a notice that the pop-up was blocked, click allow and then rerun it if you get that notification. All right, so this is a Learn Our Shiny tutorial. I think it's a really nice piece of software to demonstrate things. And what we're doing here is we're first learning how to extract the different components of multi-assay experiment, and then putting them back together to create the original representation. So as I said earlier, the different extractor functions are noted under each little schematic of the components of the class. So you have call data, sample map, and experiments. Right. So here we want to extract the experiments. So you can do experiment. So what we really wanted to provide with multi-assay experiment is a sort of user-friendly interface that is sort of intuitive and not too complicated. So if you call experiments on a multi-assay experiment object, you should get the experiment list of, so this is the list of summarized experiments. In this case, we have a few summarized experiments and a matrix in this mini ACC. So that gives you the experiment list class. right? So what happens when we use call data on a multi-assay experiment object? So let's run that. Yeah. 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 Like, yeah. Well, yeah, what we recommend is to have just one principal call data rather than multiple call data for each experiment, just to keep everything in one place. But you could store stuff in the call data. It's just that our interface doesn't really interact with the call data in those experiments. Yeah, yeah. So it'll work with single cell experiments as well, and even spatial experiments. Where the columns, you're right, right. Columns are samples, yeah. And the sample name should be tracked in the sample map. Question?
Sorry, I was just going to confirm. So samples of patients should be the rows, right? Or the columns?
For the call data, the patients should be the rows.
Yeah.
Yeah. And for the assays, the samples are the columns. So if we do call data on the mini ACC, you can see that we have our phenotypic variables or clinical variables like years to birth, vital status, basic that, so you can run survival analysis or things that you would normally do with this kind of data. And this is represented as a data frame, capital DF data frame from the S4 vectors package. So one of the convenience functions that we've added was this $sign, so you can access a column in the call data by just using the $sign and something like rate. So, and I might need to reload this mini ACC. But you can also use the call data interface, which is also a data frame, so you can do call data, any ACC, and then $sign rate. And that should work the same. So just to make it a little bit easier, we've added the $sign operator for the multi-assay experiment. And now we get to the assays, right? So assays is a special extractor function, which sort of transforms the data, not as it's stored in multi-assay experiment, but tries to give you a list of matrices. So this is helpful if you're doing some downstream analyses like TCA or ECOA or things like that. So so if you run assays, you should get a list of matrices.
So can you try to stop sharing and share again, because online people see
Oh, right,
your
right.
past video, not sharing.
Yeah.
Stop sharing and share again. Full screen.
Entire screen.
Entire screen is just another thing. Go back. Now it's good.
Okay, thanks. Wait, for some reason I need to load. Sorry? So assays is this special extractor function that gives you a list of matrices. So they're a little bit watered down, like it removes the structure that you had put in originally. So if you had a single cell experiment, it'll only give you the matrix within that single cell experiment. It'll just give you the first one. Yeah. Yeah. No, it's good. Yeah, you should be able to, or you can do just assay singular and select which one you want to extract. So not much to see here. You just see a simple list with all of the experiment names, but you can drill down a little bit and pull out maybe the stick. So because this is a list, I'm using the double bracket operator and the name of the experiment. So it should give you a matrix. So you'll see that this is just a plain matrix of statistic values. And then finally, the sample map. So you do sample map in the ACC to get a look at it. I might have to load data again. So I'll do that. So you'll see that we have the 3 columns that are part of the sample map assays for, so each experiment in the multi-assay experiment should have a name, and you'll see them listed here. And then you have your, what are patient IDs for TCGA here, the patient barcode. And for the call name, you'll have the sample identifier for each column in that RNA-seq2 genome dataset. So a little bit of trivia. What function do you use to extract the experiments from mini ACC? Experiments? Okay. Correct. Yeah, so it's easy to sort of confuse experiment list, which is the name of the class, and experiments, which is the extractors. Unfortunately, we couldn't use both to extract and to create the class, so we use the lowercase experiments to extract the data. So, and as you can imagine, the constructor function is just multi-assay experiment. So once you have all of those components, the experiment list, the call data, and the sample map, you can construct your multi-assay experiment. And one caveat, if your identifiers, your patient identifiers are the same as your column identifiers, then you don't really need a sample map. It'll get generated for you because they're the same. But if you have different sample IDs and patient IDs, then it's better to create a sample map and and feed it into the multi-assay constructor function. So we'll do multi-assay experiment and say experiments equals, and then our experiment call data equals call data, and then sample map equals the mini ACC sample map. You could I note that you can also add some metadata in case you have some things that you want to include in the multi-assay experiment, like what date the multi-assay experiment was built, or other things that are pertinent to the data. So that's also a possibility. It's a list, so you can pretty much add anything you want in the metadata slot. And if everything goes well, you should get a multi-assay experiment from this. And I think I forgot to load the data, but we'll see if that works. Yeah. So you get a multi-assay experiment, just like the one that we started off with. And that's how you would build your first multi-assay experiment from scratch. And then we have another tutorial under the cBioPortal data. So if you go under tutorials, cBioPortal, and then click on the cBioPortal data R&D and run the document, it'll give you an intro of how to use the package. So there are two main ways to use the package. One way is to download all of the data that's stored on the cBioPortal data. The project uses Amazon, so all of their data is on a Amazon bucket. So what you could do is download the tarball of all of their data and bring that in and create a multi-assay experiment out of it. If you're looking for a more fine-grained approach to the data, you can use the main function, which is cBioPortal data, and that will allow you to select sort of what assays you want, what patients you want, what set of genes you want from that particular data set. So those are the two main ways, and maybe I can show you the cBioPortal data vignette. I'm going to go to help, package equals cBioPortal data. Go to user guides and package vignettes. So we'll go to the user guide, which is the first link, and so if you're using the API to pull in the data from the cBioPortal website, you would use something like this. So first you create an instance of the API, right? So the API is sort of a living, breathing thing. So it needs to get updated every now and then. If the cBioPortal team changes the API, then the package has to also change. So you create the cBio object, which is an instance of that API, and then you use that to run it through say, the get studies function, which tells you what are the studies in the cBioPortal project. And that will give you a nice table of all of the data sets they have. Some of them are TCGA, others are not. So they have about 360 studies. So if you're interested in browsing through those studies, you can run this command and get a list of all of those studies. So the first main function is the cBio data pack, and that will pull in the zip tarbles. So you only need to give it a study ID, and that you would get from the get studies command up here, right? And you'll see that there's a study ID column in this small table. Once you have the study ID that you want to download, this one we're using AML, leukemia data set from TCGA. You do cBio data pack, the study ID, ask equals false, and then it would give you all of the data in that tarball as a multi-assay experiment. So you see that we have copy number alteration data, some HG19 CNA data as a ragged experiment. You also have methylation, mRNA, and mutation data. So there's a ton of data to work with there. So you have in total 12 different assays in that leukemia data set. So then the other one, the cBioPortalData, is the fine-grained approach. And you can either specify by gene symbol or entrée ID. And you can do study ID. In this case, we will do ACC. And you can use a gene panel as part of, so the cBioPortal project has gene panels, but you can also give it a list of genes that you're interested in as input. And some molecular profile IDs correspond to the assays that are available in that data set. So there are functions to learn all of those things, like those tags for the assays, and you can see them in the rest of the vignette. But once you have a call like that, it's pretty straightforward. You have one call, and it gives you a multi-assay experiment with some data in it, so that you can work with. And that's pretty much it for the cBioPortalData. I'll be on time. Okay. No, if anyone has questions, we can. So I know it's a lot. I think this is like 8 years of work, so I don't expect you to like learn it in an afternoon. So feel free to ask questions. Yeah. Yeah. I'm sure there are, but we're not really aware of them. Or if you can, most of the experiments you can represent as a summarized experiment, or more recently, like spatial experiment. So if you can put it in a square matrix format, then it should work. Yeah. Yeah. One thing we don't work with currently are images. That's another thing that we could add functionality for. But multi-assay experiment is sort of the foundation, and then people can create specific implementations for the data. Like if you only want a spatial multi-assay experiment, you can create that class, and it'll work with the data within those spatial experiments in a more structured way. Yeah, hello. Right. Yeah. Yeah. So metadata is a list type, unstructured. But if you have a more specific set of experiments that you're working with, like spatial experiments, you can create your own class and then work with like the image coordinates for every spatial experiment. So yeah, I guess the question is, should I extend multi-assay experiment or should I just use the the metadata slot in multi-assay experiment? Right. Right. Yeah. Yeah. You can put it in there, but you'd have to like sort of access it manually and bring it out. Sorry. Yeah. I would recommend it to be an extension because if your extension is expecting that every assay has that reduced dimension slot, then you can work with that and sort of combine that or do what you need to do with it. Yeah. Yeah.
All right. Well, we are ready to our time. So if you have any other questions, just Google Marcel's name, Multi-assay experiment. You will find him. And thank you for coming. And
Thank you, everyone.